This work was funded by the Intramural Research Program of the National Institute of Allergy and Infectious Diseases of the NIH. Now, who was the very famous director of the National Institute of Allergy and Infectious Diseases? Hey guys, JDR Exospace here. So I want to introduce you to Jean-Michel Clavedier, and that is the best French pronunciation you are going to get out of me. Jean-Michel Clavedier is an emeritus professor of medicine and genomics at the Aix-Marseille University School of Medicine in Marseille, France. And he is currently trying to dig up what's known as zombie viruses, pathogens located in the permafrost of the Arctic that have been locked away for thousands, tens of thousands, maybe even hundreds of thousands of years and not known to the human population for a very long time. And what happens in the Arctic is the very thin topsoil only ever gets warm enough for a very short growing season. And so there's a thin layer of growing plant matter on the top, and everything below it in the soil is intermixed with water and it stays permanently frozen. But as the climate is beginning to warm, this permafrost is increasingly starting to thaw out. And Clavedier's theory is that as the permafrost starts to thaw out, these zombie viruses are going to re-emerge in the human population and perhaps be very deadly. Now, Clavedier is famous in the scientific community for discovering what's known as giant viruses, and this is one of them here, essentially this thing that looks like a microscopic eggplant parm. But in recent years, he's kind of taken on this new passion where he's going into the Arctic and bringing back viral samples from the permafrost, reanimating those viruses, and in a pretty classic example of a self-fulfilling prophecy, bringing them back to life to prove that they would be deadly to humans, and then writing papers about it. And in 2014, he managed to revive a virus from the permafrost, making it infectious for the first time in 30,000 years by inserting it into cultured cells. They say for safety, he's chosen to study a virus that could only target single-celled amoebas, not animals or humans. When he did it again in 2015, Clavedier and his team isolated several strains of ancient virus from multiple samples of permafrost taken from 7 different places across Siberia and showed that they could each infect cultured amoeba cells. This is the vessel that he used to bring the samples back from Siberia, and you can tell this is state of the art. And I found an article of Clavedier's that he wrote himself, and in it he describes his own research, why it's important, and why we should all be worried. And he says, although limited to amoebas, these first results demonstrated that it was possible for viruses to remain infectious in ancient permafrost since prehistorical times, a conclusion most likely extendable to viruses infecting other species, such as animals or humans. This is when the popular notion of a zombie virus was born. Since then, we revived 7 additional amoeba viruses from ancient permafrost, now pushing the limit close to 50,000 years ago, overlapping with paleontological evidence of human occupations in northeastern Siberia. The latest metagenomic studies have identified genomic traces of pox viruses, herpes viruses, asperviruses, all of which are well-known human and vertebrate pathogens. The scenario of an unknown virus once affecting Neanderthals coming back at us, although unlikely, has become a real possibility. Okay, now at this point you might be asking yourself a fair question, which is, what is the real likelihood that something is going to thaw out of the ice somewhere in a remote place in the Arctic and create another pandemic? And the answer that I have for you is, it actually may be a lot more likely than you think, and it may be already happened. Now, you may have heard of anthrax before because terrorists like to get a hold of it once in a while and use it as a biological weapon. But what you may not know is its more common origins in livestock. You see, anthrax is actually a common bacteria that's found in the soil that livestock and wild animals consume on a regular basis and sometimes can pass on to humans. And when humans get it, it can be some pretty nasty stuff. I mean, if you look at some of the images from anthrax infections, the sores that are produced are pretty nasty. And that's why it's common practice today to immunize our livestock so they don't get anthrax and pass it on to us. And we also immunize a lot of wild deer populations as well. Well, in 2016 in Siberia and the Arctic, the permafrost actually started to thaw out, and a reindeer who had previously been infected with anthrax, who had died decades ago and been buried in the permafrost, thawed out, and then that anthrax came back to life and infected local reindeer populations. The bacteria were anthrax, and more than 75 years ago they killed a reindeer. The carcass got covered in a thin layer of permafrost, Russian officials think, and for decades it lay there frozen. Then that summer, a heat wave hit and a thicker layer of permafrost melted, and the reindeer's carcass rose to the surface. As it warmed up, so did the anthrax. Infectious spores spread across the tundra. Reindeer grazing nearby picked up the disease. And then it got passed on to people, and that's the nasty part. A 12-year-old boy ended up being killed from this outbreak, And the boy was one of 72 nomadic herders, including 41 children that were hospitalized after reindeer began dying en masse from this anthrax outbreak. 5 adults and 2 other children have been diagnosed with a disease known as Siberian plague, and it hadn't been seen in Russia since 1941. More than 2,300 reindeer died, and at least 63 people were evacuated around the site of this outbreak. Now, the crazy thing is that this was actually a lot more deadly for the reindeer population than it was for humans. There were about 2,650 animals that were infected, of which 2,350 died, and that is a case fatality rate of nearly 89%. So almost every single reindeer that was infected with this anthrax ended up dying from it. And scientists are really worried about events like this because it's not just direct contamination from things like anthrax. There's all kinds of frozen microbes, bacteria, viruses we're not even aware of that could thaw out and eventually make their way into the water supply as the ice thaws and these move downstream. Now, this is where the story starts to take a little bit of a surprising turn because scientists are actually taking a much more proactive role at reanimating some of these ancient dead viruses than you might think. You see, in 1918-1919, the Spanish flu, a type of influenza, swept across the planet, killed 50 million, 100 million people. We're not even still sure to this day exactly how many people died from this influenza. And it spread to even the remotest places of the world. And one of those remote places was an Inuit village in Alaska called Breivik Mission. And that small town had so many people die of this flu that they eventually ended up putting all of the bodies in mass graves. And in the 1950s, this group of scientists got the bright idea, because clearly they hadn't seen any zombie movies yet, to dig up the bodies from this mass grave, and that way they could recover samples of this 1918 influenza and take it back to a lab and study it. Now, in the 1950s, they weren't very successful. They were not able to recover any samples of this 1918 flu. But in the 1990s, a few decades later, a different group of scientists got the idea to go back once the permafrost thawed out again and dig these bodies back up. And they were successful. And it actually wasn't until several years later that some of the fruits of this research began to manifest as genetic science caught up. And I wanted to share some of that research with you because it takes a surprising turn. Now, there's several different types of influenza viruses, but the ones we're most concerned about as humans are influenza alpha viruses. Human influenza infections caused by alpha influenza viruses are the only members of this viral family known to cause pandemics and to be associated with high mortality. And this research actually revealed some really surprising conclusions about influenza A viruses. Mainly, that the 1918 influenza A virus was a new founder virus that initiated the current era of circulating influenza A viruses by evolving into progeny pandemic viruses through genetic reassortment. So in effect, the 1918 virus was the granddaddy of all influenza pandemics that humans have experienced since then. These include not only the antigenically drifted descendants of the 1918 H1N1 virus itself, but also the genetically reassorted pandemic viruses that appeared in 1957, 1968, and 2009. And it shows a graphic here that shows the number of deaths from these different major pandemics since 1918. And you can see 1918, very deadly, greater than 50 million deaths. 1957, this Asian flu comes along, 1 to 2 million. 1968, 700,000 deaths. 2009, the swine flu, 363,000 deaths. But there's some important details here that are kind of buried in this research that they're not making a case about, but very relevant to today. You see, it says here in the description below this figure, in 1977, pre-1957 human H1N1 viruses reemerged, presumably through accidental release of an older human strain from a laboratory. Now, you see, this really got me thinking, because not only is this relevant for today, but there were some very specific claims made in some of the articles that I was reading researching this video. While there is a tiny danger that scientists resurrect dormant diseases buried underground, many researchers believe it's worth the risk to better understand the origin of deadly illnesses. And they say the threats are remote when they take the right precautions. In the case of the Spanish flu, for example, the viral fragments were essentially dead. They could only be grown in a lab. Now, I have some serious problems with that claim, that the risk associated with digging up frozen viruses in the permafrost, bringing them back to life, and then genetically manipulating them to see just what they can do may not be as safe as we think it is. Now, I don't typically use Wikipedia as a source of information for my videos, but this was the closest thing to a complete list of biosafety lab incidents that have occurred over the last several decades. And as you can see, the list of these incidents is a lot longer than scientists like to admit it is. 1903, Bacillus malae. 1932, B virus. 1943, scrub typhus. 1960, foot and mouth disease. Smallpox, Marburg virus. Smallpox, Ebola, H1N1, smallpox, foot and mouth disease. Anthrax, Marburg, anthrax, West Nile, anthrax, SARS, Ebola, foot and mouth disease. TB, tuberculosis, H2N2, anthrax, foot and mouth disease, Brucella, Q fever, Ebola, Ebola, classic fever, cowpox, dengue, anthrax, H5N1, anthrax, smallpox, Borrelia, Ebola, dengue, Zika virus, Brucella, Ebola, prions, Brucella, SARS-CoV-2, and polio. Now, as you just heard, that's a pretty long list of incidents that happened because of lab leaks, because we were studying these viruses in a lab, viruses that we pretty much already eradicated. So I still have a little bit of an issue with claiming that all of these practices are safe if they're conducted safely, which it seems like they are not always conducted safely. That being said, there is some very valid scientific research that's been conducted from pulling these viruses and studying them in a laboratory setting. And if we go back to this paper about the 1918 influenza and what they learned from it, they actually learned a lot. Intrinsic evolutionary and adaptational determinants of influenza virus pathogenicity. Viral adaptation to new hosts. Infectivity. Among the most important properties associated with influenza A virus host switching is the ability of a virus to infect cells of the new host. Pathological features of the 1918 pandemic influenza virus. But here is where the research starts to get a little, maybe they should be doing this kind of stuff. Viral factors contributing to pathogenicity of the 1918 pandemic influenza virus have also been sought in experimental animal infections using chimeric influenza viruses containing one or more genes from the 1918 virus. Now, what does that mean in layman terms? It means they are deliberately infecting animals with chimeras, combinations of viruses, influenza viruses, some pieces from the 1918 flu virus, others from other flus, and deliberately injecting them into animals. Are we deliberately trying to create new pandemics from some of the deadliest flu viruses known to man? And another interesting thing I noted when I was reading this article is that it provides its funding source. And if you click over here, you can go right down to funding and they tell you who exactly funded this research, and it's from a familiar source. This work was funded by the Intramural Research Program of the National Institute of Allergy and Infectious Diseases of the NIH. Now, who was the very famous director of the National Institute of Allergy and Infectious Diseases? Anthony frickin' Fauci. Now, I don't bring this conclusion to you to try to imply that Anthony Fauci was directly responsible for the engineering that was done on this 1918 influenza virus. I would actually say that he probably knew very little about this specific research project, simply because organizations like the NIH, the NIAID, are just too large of an organization, too large of a bureaucracy for Anthony Fauci, the director, to be in the know about every single project that they're responsible for. But I am highlighting this for you because I think it's important to understand that these organizations that are funding research around the world and are largely responsible for genetic manipulation of very deadly viruses are doing so with our taxpayer dollars, and I don't necessarily believe that adequate precautions are always being taken at every research lab where these types of studies are being conducted. And there's definitely value that's being derived from some of these studies that's undeniable. We've learned a lot about viral pathogenicity from studies very much like this one on the 1918 flu. But my question then becomes, are we actually taking the proper precautions to ensure that these viruses that wiped out millions of people don't make their way into the general population through silly accidents in a lab? And the reality is that despite what a lot of these scientists in these government organizations funding this type of research want to admit, the likelihood of a lab leak scenario is a lot higher than we actually like to think it is. But let me know what you guys think in the comments below. Do you think the scientific value that we're getting from these types of studies is worth the risk? Or do you think maybe we need to take a step back and reevaluate what we're doing here and maybe just slow down a little bit? But anyway, that's all I have for you guys. If you haven't hit the subscribe button yet, what are you doing? You're killing me. You're killing me. Hit the subscribe button. Hit the like button. Hit that notification bell so you know when I post new videos, and I will catch you guys on the next one.